Welcome, everybody. We'll just wait a minute or so to let people join before we go ahead and get started. While we wait, I'll actually go ahead and put up the um, both the uh, QR code for the mailing list in case that this link was shared with you and you want to join the list, and also the link for speaker nominations if people have ideas for future speakers. All right. Well, today, thank you everyone for joining. We have two great speakers. First up is Tyson Graber from the Children's Hospital of Eastern Ontario Research Institute. I think he's going to be talking about finding some information on host response in wastewater. I'm very excited to hear about that. And second up, we have William Chen, recently joined Jordan Petra's group at Yale University as a postdoc. We'll be talking about some of his PhD work validating wastewater-based epidemiology models using fecal indicators. So just as a reminder, how this works with the QR codes up there in case you didn't get a chance to scan them the first time, speakers will talk for about 20 minutes, and then we'll have 10 minutes for questions. If you have any questions, you can put them in the Q&A box. And with that, uh, Tyson, you can go ahead and share your screen and get started when you're ready.
Okay, thank you very much. I'll see if the technology cooperates this morning. Um, okay, hopefully I'm sharing my screen with everybody.
It looks great.
Great. Thanks very much for the invite. And thanks for joining today. I'm coming to you from Ottawa, Ontario, Canada. This is a picture of downtown Ottawa, and this was actually taken last year, I think, but it probably pretty much looks like this today. Winter has arrived early. Holiday lights are out. You can see the Gatineau Hills in the background, and the Ottawa River flows through Ottawa, as well as a few other rivers. Water is very important here. So I'm an associate scientist at the Children's Hospital here. So it's a pediatric trauma center that serves most of Eastern Ontario and some other parts. And very excited to share with you today work that we've been doing for the entire pandemic, but specifically having to do with host response that was published fairly recently. So the CHIO Research Institute is attached to the hospital, and we're about 300 or so researchers, but only about a dozen of us, including myself, are fundamental biomedical research scientists working at the wet lab benches. And some of you might be familiar with Ottawa's wastewater surveillance project. It's been going on since early 2020, March 2020. We've been recording exposure to infectious diseases since then, so it's 6 years now, going into the seventh year. Rob Delatola has been leading this kind of from the start. So he's based at the University of Ottawa. And Ottawa is about 1 million people, a little more than that, Seward, and deals with a lot of flow. So about 350 million liters a day go through the central water, wastewater treatment plant. And Rob takes the primary sludge. So the solids is the mother liquor here, and he takes really the equivalent of about 250 microliters from that entire flow. Used to be on a daily basis, now it's a weekly basis, and does everything that most of us on this call are very likely familiar with. Does N1 and 2 qPCR as well as Peprovirus as a normalization control. And we get a beautiful trace. And we have that trace again since the beginning of the pandemic, which is pretty cool. Very valuable data. This data is open. Rob publishes this as open data, and it's on GitHub for anyone to look at and analyze. So I put out these plots through my Blue Sky handle. So if you want to check it out, please go ahead. There's also other targets. So influenza A on the bottom here, and this is just looking at across different seasons. And right now we're in red for 2025. And as you can see, It's coming in quite early and quite strong, which fits with the clinical picture. We've added to this process over the years. We've learned a lot, as I'm sure you all have, and the whole community has learned a tremendous amount of information. Um, Part of that, improving that process is by measuring accuracy, finding ways to measure accuracy kind of in almost in real time. And we've published recently, coming up with a metric, a few different metrics. And one of these is just a simple kind of Z score, so you have a number of deviations away from what you would expect if both N1 and N2 were working as they should as an average. And so a negative deflection here means that you might be underestimating N1. And underestimation might mean that there's a mutation coming in on N1. And I'm just highlighting this to show that I've put together an app here, which I've been meaning to do for some time now, but just had time recently to complete. And this is posted now as a Shiny app. You can follow it at this URL here on the bottom. And this actually maps this metric over time against what's happening clinically. So clinical genomic sequencing, or you could use wastewater genomic sequencing, looking across those two different PCR loci and looking at mutational burden. So this chart here in the middle shows you the average number of mutations accumulated across those PCR loci, and you can drill down into this so it's more granular and look at, the primer and probe areas. And just a warning that new variants coming in, there's an additional mutation in the forward primer of N1. So it's kind of a good quality control measure to make sure that we're capturing an accurate measurement. Okay, my lab at the beginning of the pandemic, when Alpha came in, when the new variants came in, we put out a new assay to be able to actually use qPCR to measure the prevalence of a variant coming into a community through wastewater. That was published back in 2021, and we did follow several variants coming into the community, and this was well before maturation of genomic sequencing as applied to wastewater-based epidemiology. And then more recently, we've actually gone and been able to directly detect RNA and DNA targets in wastewater, independent of PCR, so no amplification needed. And we didn't formally show this, but this might be of interest for people who have maybe PCR inhibited samples. They might want to consider this technique to be able to accurately measure targets. There's some other advantages to this. You can parallelize it, so you can add up to 700 different targets. So for a breadth of information, it might be kind of an in-between between qPCR and genomic sequencing, which is a little cumbersome and requires a lot of infrastructure. Okay, let's go back in time again to the beginning of the pandemic, spring of 2020, when it wasn't super obvious that RNA was the best marker for us. Another marker, protein, i.e. SARS-CoV-2 protein, instead of SARS-CoV-2 RNA, we felt might have been a better marker. I could go on for this. We for another hour. We tried very, very hard, but it was clearly RNA was the winner here. But that doesn't mean that proteins in wastewater are not of interest. And around the same time, work by the Barcelo group, experts in mass spectrometry, they actually looked at peptides in wastewater, but mapped them not to bacteria, et cetera, but to human proteins. And what they found, a lot of these peptides were mapping to immunoglobulins, so antibodies. And so that was the light that went off in the head and like, maybe we should start looking at antibodies, the host response to SARS-CoV-2 in wastewater. Humor me for a moment. This is a granola bar. Think of it as wastewater. We can know some of the ingredients. We can target those and detect those ingredients. Even detect some things that we don't know about that we haven't seen before through kind of unbiased mechanisms or techniques. That's our exposure to different things, but what is happening in the organism? What is the host response? when those exposures actually enter the body. That is something we can't really derive from wastewater. So here's just kind of maybe a bad analogy of peanuts causing an allergy in a person, and you get a syndrome and signs and symptoms of anaphylaxis. Traditionally, this has been kind of dissected and looked at through clinical means. So I want to shift your perceptions during this talk from wastewater-based epidemiology as a recording of what a population is exposed to a recording of what that population responds to and how. And like I said, this has been traditionally done in the clinic. So through seroprevalence surveys to look at targeting antibodies, to look at antibody prevalence, and also cohort studies to look at other kind of phenotypes or functional outputs. I'm not going to go through all those in detail. All this to say that this gives us a deeper mechanistic understanding of disease and the dynamics of that disease in the population, and that this really goes towards identifying new therapeutic avenues. And so we see it less as a diagnostic tool and more of a functional tool in identifying new therapeutic treatments. So again, I want to shift this perception during the talk from a diagnostic platform that wastewater-based surveillance is today to a more of a functional platform. Okay, and the low-hanging fruit here, as I alluded to earlier, what is antibodies. So you may not, the group may not know a whole lot about antibodies and immunology, extremely complicated, but this is kind of the 101 of it. Antigen-specific antibodies, are they in wastewater? If antibodies were measurable and functional, and in this case, function means capable of binding to an antigen in the body or wastewater, what antibody class, so IgA, IgG, IgM, et cetera, what antibody class would we want to target or would we expect to target? So important thing to know is that antibody production in the human body is compartmentalized. Okay, so you have circulating antibodies, which are generally of the IgG class, and we're familiar with these through vaccination. IgG increases, and those are actually correlates of protection from disease. So they prevent kind of severe disease, right, when those are increased. They do that through a number of different mechanisms. Also in the body is mucosal surfaces exposed to the environment, large surface area in the body, and the way our body protects those surfaces through exposures to the environment, like pathogens, is through secreting IgAs. Okay, this class is a bit kind of cool. So they're actually monomers of IgA bound together as dimers, and this is mediated through a joining chain, so JC, and another proteinaceous material called secretory component. So together, this adds additional binding site for antigen, and it serves as a kind of a shielding mechanism from the harsh environment. And so actually people shed a lot of IgA every day. About 3 grams is about a teaspoon from your mucosa every day, including the gut. About a tenth of that is actually IgG, and that's mostly through transudation, which is the leaking essentially of IgG from the circulatory system into the gut. Although the gut can produce secretory IgG as well. As I mentioned, this secretory component, this proteinaceous material around that surrounds IgA, and as well as sugars around the secretory IgA glycosylation, protects secretory IgA or SIGA from proteases. Okay, and importantly, antigen binding, which happens on the variable ends of these antibody chains, is actually more resistant to changes in pH with secretory IgA. Also importantly, secretory IgA goes up with infection, and specifically SARS-CoV-2 infection, which is shown on the right here. This is a pattern of a breakthrough infection after immunization. In red here, you see an increase, a substantial increase in secretory IgA secretion. Okay, so does this actually happen? Can we actually detect these functional antibodies in wastewater? We did a preliminary experiment with ELISA to detect these targeting antibodies against the S protein of SARS-CoV-2 in wastewater. and in stool. So when we looked in stool, we had a COVID-19 patient in hospital, and we took their stool, and we found that we saw a signal above background. And we compared this with healthy volunteer stool, was not suffering from COVID-19, not vaccinated, and therefore should not be expressing any antibodies against SARS-CoV-2, and we saw a lower signal. And this is just a titration curve. We did the same thing in primary sludge from the wastewater treatment plant in Ottawa at early time points in green here. This is again a titration curve. Early time point in green, and in purple at a later time point post-vaccination in the community and when prevalence of SARS-CoV-2 was a bit higher. And this was just preliminary data, but it was very encouraging. So we did our due diligence and wanted to know, well, was the fraction we're looking in, was that the right one? I.e., where can we find the most protein? Because we expect to find the most immunoglobulins in the fraction that had the most protein. Long story short, and it's probably no surprise to a lot of you, more proteins are in the solids fractions of wastewater. So whether you look in influent solids, settled solids, or wastewater sludge solid components, the most protein is here. We showed this two different ways. We also looked at building level, and as you can imagine, it's more variable there, but it correlated with with solids content. The more solids you have, the more protein you have. Seems obvious in hindsight, but may not be obvious to all. In essence, this looks complicated, but it's very simple. This process involves a little bit of detergent in a buffered solution to break up those proteins a little bit. Okay, next we wanted to look at the proportion of antibody classes in wastewater solids. We looked in sludge solids and influent solids over different dates using human plasma as a control. This is a Western blot or an immunoblot targeting on the left here IgGs, total IgGs, not antigen specific, just total IgGs. And we see what we expect is a specific binding, and also for IgA. And this is semi-quantitative. You see more IgA here, but we wanted to really do this by quantitative ELISA as a complementary assay. And we can see here down in these bottom panels, more IgA, in fact, an order of magnitude more IgA in sludge solids and influent solids versus IgG. And we also looked at IgM classes. We also looked at archive samples, and this is not the best experiment because it's not paired with the same date, but we do believe that IgA is slightly affected in terms of it's less stable after it's been frozen. Those wastewater extracts, sorry, not extracts, these are raw wastewaters samples that have been frozen and then taken out of the freezer at a later date and tested. As I mentioned, secretory IgA is very important. We looked at ELISA that is specific for that larger complex, macromolecular complex of secretory IgA. And we compared that to total IgA levels, and we saw about the same amount of both. And that led us to conclude, after we did some convincing, we tried to convince ourselves that this was showing that the bulk of the IgA in wastewater solids is actually in the secretory IgA form, and in fact, that macromolecular complex. We wanted to show that a different way, and we looked at mass spec. And again, a lot of data here. The take-home message is that we found secretory component, that important part of secretory IgA. We found J chain, that joining chain, and this is in this pictogram down here. Everything in blue we found by mass spec. Plus the joining chain and the secretory components. So components of IgM, components of IgG, but we did not find heavy chains associated with IgD and IgE, which makes sense because those tend to happen in acute inflammatory conditions. So we didn't expect to see a lot of that. And plus these are normally very, very low relative to these other classes in the human body. On the right here is just additional evidence suggesting that J chain is not by itself in the wastewater. It's actually associated with this entire macromolecular complex. So all this kind of supports evidence that we are actually looking at functional secretory IgA in wastewater. The question is, and we actually saw variable chains by mass spec. Question is whether they are still competent to bind antigen after having gone through the human body and having gone through the pipes and end up ending up at the wastewater treatment plant. So we looked at this specificity for antigen, looking at SARS-CoV-2 spike uh, IgA antibodies by ELISA. And this was actually specific for the trimeric form, which sits on top of the virion and binds um, cell receptors. Um, this changes over time with variants coming in, so this might affect the sensitivity of the ELISA platform. But we did look in stool, and we saw a specific signal above the red threshold here, not IgG, which is consistent with the clinical picture. And then we looked in the wastewater, and we saw both spike IgA, and this is just a titration, and then spike IgG as well, in several different dates. We deconvoluted that a little bit to look at time trends, and it's not a strong time trend, I will say, but looking at earlier times, and this is looking at the Ottawa community, SARS-CoV-2 RNA by wastewater, you can see that prevalence is increasing over time for the dates we chose to sample in orange. And also, this was the time of a booster campaign where 25% of the population by January of 2024 was boosted. So we thought we would see high amounts of IgA and IgG against spike at these dates. And you can see, if you see in white, is below threshold for both sludge and influent solids for IgG and IgA in Ottawa, versus the later time points, you see a bit of an increase. So more of a proof of principle, not really clear trends, but it was very encouraging. Okay, I know I'm running out of time, and I'm going to put up this very complicated slide, and people are going to get a little annoyed. I want to show this because a very important question we wanted to answer was whether or not, okay, so those antibodies go up, antigen-specific antibodies go up, but do they truly report protection from infection or disease? So in other words, those wastewater antibodies, are they simply indicating exposure to disease, or are they actually showing protection from infection and disease? And this is not kind of obvious or intuitive, because I mentioned before that these antibodies are compartmentalized. All this is going to say, this is one study, a supplemental figure buried. These are 16 unvaccinated individuals, and looking at binding capacity by ELISA of different antibodies against SARS-CoV-2 S protein in different compartments. And we know that serum IgG is a very good correlate of protection against disease severity. More of it you have, the better protection you have. And this correlates with stool IgA, very important. It doesn't correlate with IgG from the stool. So there's reasons for that. I'm not going to get into it, but this is very good evidence in my mind that what we are seeing in wastewater in terms of specific antigen binding antibodies is indicative or a proxy of protection from infection and disease in the community. Okay, there's a wealth of information stored in wastewater, and we're chipping away at that. We were looking at extracting all the B cell epitope information, so the cells that kind of produce antibodies, and we're seeing a lot of functional classes showing up, including RSV epitopes. And this tells us that maybe we can look at epitopes in the population very easily, and that is important in vaccine design. And we're also looking at extracting actually T cell epitope information in wastewater. So exciting things to come, hopefully, if there's money coming through the pipe, so to speak. And I'm going to stop there. Key takeaways here for people who are watching this later on. I'm not going to go through them. I'm just going to have my acknowledgement slide, and thanks very much to Dr. Sean Stevenson in my lab, who's a postdoc, who's did all 90% of that antibody work, which is great. Shout out to the Delatola lab as well, and Ottawa Public Health and Water Services, who've helped us throughout the years with these collaborations. And thanks very much. My contact information is on this slide, and happy to take questions.
Great. Thank you so much, Tyson. That was great and a lot of information. So just a reminder, if you have any questions, you can go ahead and put them in the Q&A. I'm going to start with one that Ali actually asked in the chat, because we can't head it to the Q&A, and I had a similar thought. You make these measurements sound really easy. What are the key sort of challenges in doing these measurements, and is it possible to expand them for other high circulation viruses like a norovirus or an influenza?
Yeah, protein definitely a different story than nucleic acids, especially antibodies. Lots of issues with specificity, selectivity. So the short answer is it depends. But I think we've come up with processes to at least have a good place to start. And it really comes down to getting good kind of monoclonal antibodies. And some of those are harder to get than others. And hopefully during the pandemic, not hopefully, we have, I don't know what it's like in the US, but in Canada, we've developed processes where we could easily ask for the development of certain monoclonal antibodies. So I think those pathways to and processes to develop those tools are in place. So it's not super hard, but it's certainly not as easy as designing a probe or primer and saying this will work. There definitely is more kind of QC involved in it. But yes, it can be expanded to essentially anything with an exposed epitope
Yeah,
in the solution.
great. Here in the chat, we have a question. Can we distinguish IgG produced by vaccines and IgG produced by infection?
Yes. Short answer is yes. We didn't show this. So normally in a seroprevalence survey in clinic, you would distinguish that via looking at nucleocapsid versus S protein. So nucleocapsid would be indicative of a natural infection of SARS-CoV-2, whereas S, as we know, we're vaccinated and with an mRNA expressing S protein, hopefully. And that would be picked up by a different antibody. And so you could distinguish those two. And that's going to be different depending on, the target of interest. But short answer is yes, you could do that. We did try looking at nucleocapsid and wastewater. Unfortunately, this speaks to the specificity concern. The ELISAs we had weren't great. So those, we kind of really need bespoke tools, which requires more research.
So I'm going to follow up on that with my own question. With the specificity issue, I mean, I'm not familiar with these methods, but what is sort of the control? If you're getting a detection from matrix as complex as wastewater.
It is difficult for sure, especially since you may not have a pre-exposed population
Right.
control versus post. And if there's an issue with frozen archive material affecting that signal in the first place, definitely an issue. There's different ways you can approach that. One of them is to try to titrate away that signal and then go in with it. So titrate away with one antibody and then use one antibody as a sink, essentially to pull away that target. And if it's really specific, you should be able to come in with another antibody and still show the absence of that signal or the return of that signal. So I mean, there's a couple of different ways you could approach it. None of them terribly easy
Yeah, right.
in this complicated matrix, but it is possible.
Great. There is one more question in here about the relationship of this work to AMR. Maybe I'll let Tyson answer that in the chat because I'm not 100% sure. But with that, then maybe William, if you wanted to get started and share your screen.
Uh, oh, okay. Yeah, thank you. Uh, okay,
Looks great.
okay, can you everyone see? Yeah.
Yep.
Uh, do I just start now?
Yeah. Go ahead. Get started. Yep.
All right. Well, hello everyone. Thank you very much for being here today. My name is William Chen, and I'll be presenting a topic in which I've recently conducted a research paper on titled Validation of Wastewater-Based Epidemiology Model Predictions. and the influence of supershedders and sewage dynamics using the fecal indicators, peppermint Mottle virus, and Carjivirus. The first, quick context and motivation. First, very quickly about myself. As Kyle mentioned, I'm now a postdoc at Yale University, but before that, and actually just literally a couple of weeks ago, I was still a PhD candidate in Kyle's lab at the University of Notre Dame. Here's a picture showing me as well as fellow um, baby labber Andre Padilla Aguilar, who also defends. So we defended on the same day successfully. So uh, thank everyone in the group for this. But yeah, so as a member of the baby lab, I'm pride myself as modeling guy. And throughout my work, which I'll sort of touch upon, I've done a lot of modeling work for wastewater-based epidemiology. But there's one topic specifically which I'll discuss in this presentation. As you can see, I literally published this a month ago and like a week right before I did my defense. So just quick introduction, quick background. As you all know, wastewater-based epidemiology, or WBE, is an alternative approach to tracking infectious diseases. So here's a workflow of WBE. Starts off with infected population would shed or excrete through bodily fluids like saliva, stool, and urine, the pathogenic genome target of interest into the wastewater system where they usually are collected at the wastewater treatment plant, for example, the influent. And afterwards, the samples are then subjected to concentration, extraction, and then molecular-based techniques like PCR to detect and quantify their amounts in the pathogenic genome targets in the wastewater samples. So this method is community-wide and non-intrusive. It does not require people to get tested directly. Therefore, it has many advantages, including bypassing many of the limitations of clinical surveillance. For example, it can detect both asymptomatic and mildly symptomatic cases. It is relatively cost and time efficient, and also it can identify cases days or even before, or sorry, even weeks before the clinical surveillance. So it can be of great benefit. And it has been demonstrated to be useful for tracking diseases with high fecal shedding rates, for example, poliovirus and norovirus. And then recently, as you all know, it can be used to track COVID-19 cases. Since the pandemic, in fact, there has been a significant rise in the publication and interest in WB research. And researchers are asking the question, can this promising alternative approach be applied to novel disease targets, for example, emergent and neglected diseases? And in fact, this question is what I aim to address through my PhD work. However, answering this question can be tricky because the primary and way of developing WB for novel infectious diseases is through an ad hoc or trial and error approach. This approach deploys the laboratory and field experiments before it is known beforehand whether or not a disease is suitable for WB. So if WB were to be applied to a disease unsuitable for wastewater surveillance, it can lead to wasted time, effort, and resources. And even if detections were occur without a quantitative benchmark to compare detection results to, it would not be clear what these results would mean. Therefore, it is very difficult to assess what can be done in the future to improve the success rate of wastewater detection for these targets. That is why in my PhD work, I've developed a mechanistic approach, or in fact, a framework that can help assess the WB feasibility of various disease targets, including novel diseases. And since everything in my model is quantitative, it can help provide a means to understand how to improve the success rate of detection for these pathogens. So here is the framework that I've developed. In general, the framework utilizes input values, including genome shedding rate, wastewater flow rate, limits of detection, and the reported case rates to predict output values shown on the right, including wastewater concentration, probability of detection, and minimum infection rate or optimal number of PCR replicates that can help inform how suitable and feasible a target is for WB applications. Well, I divided my framework into three major steps. The first step mainly is to predict the wastewater concentration distribution through literature review of shedding data for the disease target of interest. Once this concentration distribution is generated, it is then compared to a process limit of detection, which I'll talk a little more about later, to find the probability of wastewater detection for that target. So then I can then do the third step, which is to conduct the feasibility analysis, which includes, for example, finding the minimum infection rate to achieve this desired probability of wastewater detection, for example, 50 or 90%, as well as what can be done in the lab, for example, what the PLD value should be, as well as what the minimum or optimal number of replicates for PCR assays should be applied for WB protocols so that the target can be detected feasibly in wastewater. So I highlight on the right side here that there are many applications of my model, including what I just mentioned. I can distinguish which targets are suitable for WB. Also, I can predict when, where, and how it can be most successfully applied. So by when, I mean what extent of an outbreak is needed. By where, I mean what sampling locations are appropriate, and by how, I mean, again, what should be done in the lab. So what the number of replicates and PLD values or sensitivity of the protocol should be. Also, my model-based framework quantifies the relationship of five primary factors which affect the probability of wastewater detection. So shown in this uh, image, as you increase the genome shedding rate or the infection rate, as well as the number of PCR replicates, this will increase the likelihood of wastewater detection. Alternatively, the PLOD value can be decreased, meaning the overall sensitivity of the WB protocol can be increased, or the per capita flow rate of the sampling location decreasing will also cause the probability of detection to rise. So I use this framework to predict, or what I like to call forecast, the WB outcomes of various novel disease targets, including Mpox and Zika virus shown here. So I was able to use my model to predict that Mpox can be feasibly detected in wastewater ahead of many other publications that reported the successful detection of Mpox in wastewater systems throughout much of the world, including the US, Spain, Thailand, and France. For the case of Zika virus, though, my model predicted that it will be substantially more difficult to detect in wastewater since it has a much lower shedding rate. And instead, we'll need high amount of fine-tuning of WB protocols along with uh, sampling at locations endemic to Zika virus, for example, Brazil. And this is consistent with what's reported in literature because so far only a couple of studies have detected Zika in wastewater and at very low detection rates below 6%, for example. So in an indirect way, I was able to show in the third step that my model-based framework is can have a valid and robust prediction. However, in this study that I'm now going to talk about in detail, I want to illustrate a systematic way to validate the robustness of my model predictions. Because now I'm going to try to show the accuracy of the first two steps, which are important because the feasibility analysis hinges on their accuracy. So the first step is, again, estimating the wastewater concentration distribution, while the second step is to use that distribution to then estimate the rate of wastewater detection. So here is just a gist of what I did in this study. So I compare values predicted using the model-based analysis. For example, collecting shedding data to arrive at this conclusion, and then compare it to values derived um, using wastewater data. So to conduct this analysis too, I will also need to select a wastewater target that has both high shedding and wastewater data. And two fecal indicators come in mind: Peppermint mottle virus or PMMOV and Carjivirus, formerly known as Crassphage. So both targets are found in high abundance and concentrations in the human gut. Therefore, it is expected they'll be shed at high amounts in wastewater streams. So to do this analysis, I did two comparative analyses. The first comparative analysis I did was comparing predicted versus observed total wastewater loading or genome copies of the PMMOV and Carjivirus. So for the predicted side, I used the same approach where for novel disease targets, for using my model-based approach, by finding shedding data of both fecal indicators and applying this equation shown above. This will generate this predicted total wastewater loading of the fecal indicators. Now for the observed side, I use a different approach using the equation below, where for each study included in my analysis, I uh, recorded the total wastewater concentration values for the fecal indicators and then multiply that by the per capita daily wastewater flow rate of the sampling location where these studies were conducted. By aggregating all the data, I can then generate this observed total wastewater loading of PMMOV and Carjivirus. And note too that I've divided this product by the decay term shown here because I'm doing the comparative analysis at the influent of the wastewater stream. In other words, before any decay were to occur. So in my second comparative analysis, I compare the probability of wastewater detection predicted using my model-based approach with what's reported in the literature for the actual frequency of wastewater detection for both fecal indicators. So again, the predicted wastewater probability of detection is can be found by comparing the PLOD to the wastewater concentration distribution, and the fraction that is of the distribution above the PLOD is considered the estimated probability of detection. So as promised, here's the definition of the process limit of detection, or PLOD. I define it as the assay limit of detection, or ALOD, normalized to the original wastewater sample volume that accounts for total losses of DNA and RNA throughout the WB workflow. So in the case when the PLD is not already given in the literature, I estimate the minimum, median, maximum values using the range of recovery rates for both fecal indicators subjected to different wastewater concentration methods. So here are my results. First is the total loading of PMMOV and wastewater density plots. So the dotted curve shows the predicted density plot, while the solid curve represents the observed. So from this plot, you can see that there is generally a good agreement between the two distributions, as there is over 80% and 90% area and range of overlap. Therefore, this supports my model-based approach as a robust tool to predict the total wastewater concentration, loading, and therefore concentration of PMMOV. However, the median of the observed is greater than that of predicted by about 8.1 times. A similar conclusion can be drawn for Carjivirus comparative analysis. This time, though, the observed uh, median is greater than that of predicted by 3.7 times. So this disparity in median values is important because this can provide evidence of both the presence and impact of supershedding for wastewater measurements. So supershedding is what occurs when a small subset of the population shed at orders of magnitude higher than what is observed in the general population. Hence, they'll dominate the overall wastewater signal. Another potential explanation for the disparity could be, for example, sewage accumulation effects. For example, PMMOV being accumulated in biofilms in the sewage pipes, or Carjivirus growing within bacteria in the sewage system. But regardless of what the effect is, it is important to account for that because, as you can see from this equation, prevalence is usually estimated using wastewater concentration data by dividing the concentration by an average shedding rate value. So if this average approach does not account and correct for the influence, for example, of supershedding, this would cause PMMOV and Carjivirus prevalence to be overestimated by 8.1 and 3.7 times, respectively. So here's the result of the second comparative analysis, where I show here each of the studies included in my comparative analysis and the approach where I compare the PLOD values with the wastewater concentration distributions for each study. This is shown for PMMOV, and here is similarly for Carjivirus. So now comparing the predicted estimated wastewater concentration distribution for each of these studies with what's reported in these studies, I was able to show through the right-hand side of this tabulated results that for the most part, for both fecal indicators, there is only a 5% difference at most between observed and predicted wastewater frequencies of detection. Hence, this supports my model again as a robust tool to predict the wastewater probability of detection, at least for these fecal indicators. So I'd like to acknowledge some limitations of my comparative of this study. First off, I've only used fecal shedding to do my analysis since there are so far no study to date that reported any saliva, urine, or other bodily fluid shedding rates for these fecal indicators. Also, I'd like to highlight that the shedding and wastewater data I collected were mostly from Western developed countries. Also, for the case of shedding rate, they're mostly conducted for patients with SARS-CoV-2. So it is unclear if, for example, having SARS-CoV-2 might influence shedding of either fecal indicators. Therefore, this can create some bias in the results. And finally, when I did my PLOD estimation, there's still a limited amount of recovery data available for both fecal indicators. So in conclusion, I would like to say that this study supports my model-based framework as a valid and robust tool, while also highlighting the potential influence and therefore importance of accounting for supershedding in wastewater measurements. Now, I've only done this analysis to 2 fecal indicators, but in the future, this similar comparative analysis can be done to other targets, therefore helping further validate the robustness of my model while also elucidating what the specific effects of supershedding would be for these specific targets. So targets include pathogens like norovirus and adenovirus, or other fecal indicators like HF-183 and the recently discovered BBI-143. So I'd like to thank everyone again for their attention, and now I'm open to any questions. Thank you.
Thank you so much, William, for explaining to us the modeling that you've done. And I know it's always challenging to put together a modeling presentation. You did a great job, so thank you.
Oh, thank you.
So please put any questions you have for William in the Q&A. We have a bunch of questions here, William, and I guess I'll start in order, and I have some as well. So Nalco is asking about degradation of targets in wastewater. Is that something that you included in your modeling or not?
Yes, so I did mention one of the slides that I normalized everything by the decay rates for both PMMOV and Carjivirus. So I also did include that in my analysis. And actually, originally I was trying to compare if like effects of supershedding is more important or if decay is more important. And if you look at my paper, I did a sensitivity analysis as well as assuming that if the hydraulic residence time, which is the time it takes the target to reach the influent of wastewater treatment plant, which is about 3 hours, it generally plays a very minor role, like probably about 95% of the PMMOV and Carjivirus would still be intact, even after it decays. So from my study, it seems like supershedding is a lot more important.
Great.
Good
Okay,
question. Yeah.
there's another question about viral shedding rates. And I think that in your presentation that you indicated that you use like one value, like certain number excreted per person per time or something. But we know that shedding rates, at least for pathogens, might change over time. And I guess this person is asking also about, sorry, I'm going to sneeze. No, I didn't sneeze. Okay. Is also asking about the variability in shedding rates. Did you assume for stuff like PMMOV and Carjivirus that it was just constant? And what would you do if it wasn't constant? How would you handle that? Tell us a
Okay.
little bit
Well,
about that. Yeah.
well, great question. Can I share my presentation again?
Sure.
Yeah. So, yeah, great question again. Yeah. So I didn't assume a constant value in my shedding rate approach. What I did is I used the same approach I did for my model-based prediction with shedding rates. So I collected all shedding data from available literature to produce a distribution of potential values. So, and this includes all levels of like durations as well. Therefore, I assume that this distribution will reflect the population um, aggregate of the overall individual shedding distribution. And this will include like all frequencies of the different like onset of symptoms, or in this case, for when someone has PMOV in their stool. So like it could be a few days, a few weeks, and etc. So to answer the question, yes, I included all potential shedding rate values in my analysis.
And then if you were going to extend this to something like norovirus, which presumably is not shed by everybody at, well, it has like a very specific, like, increase, high level, and then decrease after infection. That would be, you'd have to take a different approach to modeling that. Is that right?
Wait, sorry, I didn't quite catch the last part.
Well, I guess I think that what the question is getting at is that, for PMMOV, tell me if I'm wrong, but I feel like that we understand that PMMOV and Carjivirus are kind of like intermittently excreted by everyone, regardless of disease state. And so this type of approach of developing a distribution and pulling from it seems appropriate because it probably is right for people. But when you have norovirus, it's different because because only people who are infected are excreting it. It just seems like it's a little bit different.
Oh,
And I wondered if you thought through a little bit about how your modeling might change for
yeah, so I didn't really go over this much, but in this equation, I would take that into account by multiplying by the fraction of the population who shed whatever target in the of the target of interest. So for norovirus, if only 30% or a very small fraction sh shed out, it's based on what this model is based on what ends up in the sewage system. So it takes into account how many people of the population also shed the target as well.
okay.
So it'll estimate how much, like for PMLV, it's almost 90%. So I multiply like 90%. And that also, so for regardless of what the shedding rate is for the population, that is also taken into account.
Okay.
Yeah.
All right. Thank you. I have a question. I was wondering, after you've done all this work and you're modeling, I'm sure that you sort of thought, oh, I wish that there was this data available or more of this data available that wasn't. And so if you were going to ask people to like generate more data in a certain area,
hmm
what sort of data did you feel like you would need more of to
Oh,
do this modeling?
okay. Well, thank you so much for asking this because the primary challenge of this model currently is because there's just not that many shedding data. So to answer your question, for sure, definitely more shedding data, but also more understanding of the process limit of detection for various targets as well, because many studies don't report the process limit of detection, which is not just the detection limit of the assay, but also um, fine.
Yeah.
Yeah, also all the information that's needed to estimate that limit of detection for the overall process. So
Yeah.
I can compare apples to apples, like the
Yeah. And that's
wastewater concentration and the limit of detection normalized by the wastewater sample volume. But also recovery rates is also significantly under reported, not only for these fecal indicators, but for actually many pathogens in which I try to
Mhm.
attempted to do this what model-based approach. So great question.
Great. Thank you. I'll put in a plug for the ME guidelines, which actually ask people if they follow them to report this type of information so that you have it. Um,
Yes, shedding data for sure, number one. That's if there's no shedding data available, I can't apply this model at
yeah.
all. So yes,
Okay, great.
yeah.
We're out of time, so we won't be able to ask the additional questions. Thank you so much, everybody, for joining. And then I'm pretty sure we won't have a webinar on January 2nd because although it's on the calendar, that's still a holiday. So we'll see everybody in February, and we'll send out an email reminding you there won't be a webinar on January 2nd. We wish you guys an amazing New Year and winter holidays. So take care and we'll see you. If not sooner, we'll see you in February. Thank you.